Hello, friends. Welcome to BioBunk once again, and this is our medical block section. So, in this section, now we have taken a topic called interhemispheric brain inhibition, and it's an interhemispheric brain inhibition of human motor cortex via transcranial direct current stimulation and its advancement over others. So what the theme here is to understand first about the anatomy of brain where we are having two cerebral hemispheres, right? So there are two cerebral hemispheres. So one is right and one is left, and there pattern in, and we already know about these cortex of the brain, the several areas which is there over the cortex of the brain, frontal, parietal, occipital, and temporal one. But what we need to understand here is corpus callosum. So corpus callosum is a white myelinated fibrous sheet which is connecting. Okay, so white fibrous myelinated sheet which is connecting the brains. The right hemisphere of the brain is connected to the left one with the help of this corpus callosum. Okay, means what we have to understand here, this corpus callosum is interconnecting both the cerebral hemispheres, the right and the left. So both they are competing for centrality. So the cerebral hemisphere, so the left and the right one, basically they compete for the centrality. So like left says I want to be central and right says I want to be central, right? So this is what corpus callosum is doing. It is giving all the features, like whatever left is doing is known to the right one, and whatever right is doing is known to the left one. And whereas the interconnection, every like right and left cerebral hemispheres are connected, several fibers are there, like in case of pons, and all the fibers are getting interconnected in the region of pons as well. But 70% of them are interconnected with the help of this white fibrous sheet called corpus callosum. And among those connections, they are inhibitory in nature. Okay, so these corpus callosum are inhibitory in nature. So they check. Okay, they basically check what is the need of the connection. Like they like right. If the right is having exaggerated response, okay, so the left one is the one which is stopping this exaggerated response, and vice versa. Means if the right one is doing the exaggerated response, right is doing exaggerated response, so left is the one which is blocking that, which is inhibiting the functioning. So this is a normal physiology of corpus callosum. So the interconnection, which is of course a positive way, like inhibition is required because the over exaggeration of any of the cerebral hemisphere is not good, right? So what happens in case of cerebrovascular accident? Of course, we're talking about the stroke thing. So when suppose this when suppose this area of the brain is getting in because of hemorrhage or ischemic stroke, we know that because of the infarcted area, this particular part, suppose this right part of the brain is like right part of the brain is ischemic, or because of the hemorrhage it is getting infarcted. So right is affected. Okay, right is affected and left is already inhibiting its activity. It's already okay. So right is controlling the activity of left and left is controlling the activity of right. So what we want to say here, like in case of cerebral vascular accident, the decreased excitability in affected hemisphere, whereas the reverse is seen in unaffected side. Okay, so exaggerated response on the affected side, and there will be no more interhemispheric inhibition, right? So this condition leads to spasticity. I think everybody understand about the spastic condition. Spastic is when there is exaggerated response, and flaccid when loss of muscle tone is there. So loss of muscle tone, why there is loss of muscle tone? The exaggerated response because there is overactivity. Okay, so there is overactivity because why there is overactivity? This particular area of the brain is infarcted. Okay, so when it is infarcted, it is not inhibiting the, when the right hemisphere is infarcted, it's not inhibiting the left one. So left one is performing more and more. So left part is having an exaggerated response called spastic condition, whereas in case of left, the left one is inhibiting the right, which is already being inhibited because of infarcted. So it is inhibiting, as so there is a flaccidity, loss of muscle tone. So we understood what is happening in case of stroke. In case of stroke, what is happening? The part which is being infarcted, okay, the part which is infected, infarction is like that area is paralyzed, okay, that area is not functioning well. So when the right part is infected, left part will part will show exaggerated response because right is not inhibiting anymore. So you can do whatever you want to do. This is what right is doing in case of stroke. So if right is infected, left is feeling exaggerated and right is having a flaccid approach. So this is the thing, this is a problem. Okay. So let us understand how the problem can be sorted out with the help of tDCS, which is the theme of today's topic. So tDCS and its advancement applied over interhemispheric brain inhibition. Let us understand how it is used first. Like tap up and tap down by tDCS. Okay, we can see the device that the electrodes has been applied over the certain area of the brain. Of course, we will see by MRI scan and all. So by MRI, we will see which area of the brain is infected and where we have to apply the electrode. So this is the main question where we have to apply the electrode that we will discuss later on. But first we have to understand what is this tap up and what is this tap down by tDCS. We understood what is the problem. In case of stroke, the area which has been infected, the opposite side you will see a spastic condition, and the area which is not like the area which is okay, which not been infected, like there will be spastic thing, and the another side which is infected, there will be a flaccid thing is seen. Okay. For this, a tap up and tap down method is applied. The area which is inhibiting the exaggerated response, you have to, the inhibition should be slowed down. So we will do tap down. When you want to inhibit the inhibition, when you want to inhibit the inhibition, because you don't want the flaccidity there in your affected area, right? So the inhibition of, it is inhibiting the inhibition of by the tap down process, and by tap up, what is done? By tap up, you will like increase that affected, like in affected area. When you will do tap up thing, you will increase the activity. So in affected area, you are going to apply a tap up electrode, and in inhibited area, you are going to apply the tap down. I hope everybody understood this. So